All right, we will go ahead and get started. Good afternoon, everybody. My name is Kathy Grunling. I'm here today representing the Georgia CTSA, which is sponsoring this speed networking event, as well as the Emory School of Medicine. So today we're going to hopefully have fun in the next hour and a half and meet new collaborators. Our goal here is to connect people with each other. Hopefully you'll find someone working on something similar to you that maybe you haven't run across before, and you're able to set up a new collaboration. So we are going to talk about the microbiome and health and disease, a pretty exciting and cool topic. I'm going to set some ground rules and then turn it over to our Dean, Carlos Del Rio, who will be the MC for the day. So during each presentation, unless you're speaking, please mute yourself. You will have 4 minutes. Our Lauren James, who's running the show today, will be advancing the slides for you. In the upper right part of the screen, there'll be a small timer so you can keep track of your 4 minutes. At 4 minutes, if you are still presenting, you're going to hear a gong signaling your time is up. You want to hear the gong, Lauren? If you're still presenting, that will stop. That will be the end of your presentation. So please stick to your 4 minutes. Our presenters are in random order today, so stay on your toes. You won't know who's coming next. Next slide, please. So after each presentation, we'll have 1 minute for questions. If you have a question during that time, please use the raise your hand feature, which can be found on the bottom part of your screen. And then if there are more questions afterwards, you can put them in the chat and have a conversation in the chat with the presenter. We have created a presenter book that contains contact information for all presenters. So if you hear a talk and you're interested in following up with someone, you'll be able to do that. With that, I will turn it over to our MC. Next slide, please. Who's Carlos Del Rio. He's our interim dean of the Emory School of Medicine. He's also the Leon Haley Distinguished Professor of Medicine, the interim chief academic officer in Emory Healthcare, and an ID physician, so very familiar with microbiome. So Carlos, thanks for doing this today, and I'll turn it over to you to get us started.
So thank you, Kathy, and first of all, thanks everybody for being here. I think this is very exciting. Microbiome is, to me, fascinating. You know, we live in this environment that we share with microorganisms, and these microorganisms, not unsurprisingly, contribute to both our health and our disease. And I think understanding how our environment, our symbiosis with these microorganisms impacts everything we do, I think is fascinating. I remember, many years in medical school when I first learned about the role that the gut microbiome microbes had in synthesis of vitamin K and other things. Who would imagine that it had a lot more emphasis, more impact? Just recently, I read about potential implications for Alzheimer's disease and many other diseases. So I'm very excited to hear today's presentations. And I think what we'll go ahead and is go ahead and get started. And Kathy, you want me to start by just presenting each one of the presenters? So our first presenter is Dr. Ravindra Trabhusayam, who is an associate professor in the Department of Pediatrics, as well as an associate professor in the Department of Biomedical Engineering. He earned his MS in Biomedical Engineering in Argo, Paris Tech, and his PhD in Developmental Biology in the College of France of the CNRS. He trained in the postdoc research in human immunology and genetics at Stanford, and he joined the faculty at Emory in 2011. And his laboratory does patient and model-based studies of cystic fibrosis, TB, COVID-19, lung and breast cancer, malaria, and focusing on innate immunity and metabolism. And it's really about targeting, understanding the relationship of this through proteomics and metabolomics and doing profiling. So he's been supported by NIH, by NSF, by the CF Foundation, and I'm looking forward to his presentation. So go ahead, Ravindra.
Thank you, Dean Dario. So I think we went over the first slide. What I'm going to talk about today is our ability to analyze innate immune cells that get recruited to mucosae or to mucosae upon stress, and see how they regulate the interaction of the mucosa with the microbiota. Oftentimes we think about it as a tug of war. What we realize more often than not when we analyze these cells in vivo is that they are trying to reach an agreement with the microbes, be they fungal or bacterial microbiota. So there's a lot I think that we need to do in terms of understanding their importance in driving commensalism and tolerance. In addition to sometimes when the microbiota is dysregulated, use them as helpers to cull the numbers of bugs and essentially use the innate immune system to our benefit. So I'm going to go to the next one, please. In the next slide, you can see a few snippets of what we do in the lab. We essentially go directly to clinical samples. We analyze the cells, the fluids, and we try to collate the information from multiple omics and functional assays to figure out what the innate system is doing. So when I say innate, I'm thinking mostly the epithelium, the resident macrophages, and oftentimes the granulocytes and the monocytes that are recruited therein. What we found, especially in CF, but in other diseases as well, is we can take, for example, fluid from a patient and put it on top of a makeshift epithelium, recruit blood cells to that environment, and we find that by that simple contraption, that's combining the transmigration and the exposure to the mucosal fluid, we can basically mimic in vitro the conditions that are present in a normal or a pathological mucosa. And we can also study that way, mass produce the immune cells and understand what human immune cells that are mucosa conditioned, how they react to a particular virus, bacterium, fungus, or combinations thereof. So we can derive biomarkers from there, but we can also use these models to develop drugs further. So I'm going to go to the next one, please. And that I said already. So we're going to move to the next one. I think our opportunity here is to understand how the transmigration of these cells and their cooperation with the epithelial cells in the lumen ends up helping define how the host is not just clearing or avoiding pathogenesis caused by luminal fungi and bacteria. The next one, please. Also, how more often than not, as I said earlier, this is not defined by a constant tug of war, but more by relationships of commensalism, of tolerance. There's a lot of crosstalk that's happening between the microbiota and the host cells that are present in the lumen via metabolites, which are a common alphabet between prokaryotes and eukaryotes, as well as extracellular vesicles. Next one, please. And this, in part, is driven by the reprogramming that the innate cells can undergo very quickly when they are in the lumen of an epithelium. So understanding that allows us to develop new myeloid-enhancing means to better treat disease, but also better understand how we can reestablish a normal microbiome host relationship in some cases. Next one, please. So we do that by analyzing patient samples. Next one. We do that also by mimicking those interactions in vitro in biomimetic models. Next one, please. So we think that can lead to the discovery of myeloid-enhancing drugs. I'm going to finish in the next slide by just offering a couple of things that I'll leave you with that slide. Thank you for the gong. It was great.
Thank you very much. Let's move on to the next presentation. Aditra Mishra, who is an assistant professor at University of Georgia.
Thank you, everyone. Right. So, okay. By training, I'm a statistician. I have my PhD in statistics from University of Connecticut, and recently I joined the Department of Statistics at University of Georgia. In my past postdoctoral experience and after that at MD Anderson, I heavily focused on understanding the challenge associated with microbiome data, and then focused around developing statistical methods or computational tools around microbiome data analysis. So, and most of the time I have worked in a team science framework where I've looked for hypotheses from our collaborators, and then after understanding the challenge, developed statistical tools around it. And as far as the standard statistical framework that is concerned, I use high dimensional statistics, multivariate analysis, robust statistics, variational inference, regularization, these kind of infrastructure to deal with the statistical challenge that arise with the microbiome data. And then as far as the multiomics or microbiome host interaction model is concerned, I try to use all these different sets of tools in a very, very efficient way to come up with either methodology or tools for analyzing the microbiome data. Next slide, please. So as I said, right, so it has been a very organic approach in my research career. At a given amount of time, like I wear two caps. One cap when I'm as a consultant to any microbiome related project, I try to use existing set of statistical methodology on any collaborative project and try to set up a pipeline for analyzing the microbiome data in a very, very efficient or the state-of-art available microbiome tools. And when I am as a statistician cap, right, when I try to learn from the collaborator, if I get a new hypothesis for which there is no computational tool, that's where my methodological expertise comes in. So these have been my two different approach through which I've been serving microbiome data analysis or in general, life science domain in general. And so far, some of the methodology that I have developed over the period of time has focused on some of the areas like genomics, microbiome for sure, health study, neurological science, and so on like that. As far as like the basic idea, when we are now thinking about microbiome data sets, we have already thought about like, okay, there are individual species that have been doing all the job. But now as we are converging more towards microbiome and its functional role and how microbiome as a community act together, some of the methodology that I have developed over the period of time, especially in the framework of multivariate analysis, when we are trying to integrate microbiome data with other omics data sets and so on like that, tools like statistical methods for multivariate analysis, where I try to encode the complexity of the omics data sets in a general framework, and then I try to some of the methodology that I have developed over the period of time, and have been working on some of them with the new. As I identified new problems, is focused around that. And as in my standard multivariate analysis tools, I try to use low rank and co-sparsity structure, which kind of enforce in a data-centric way and enforce the microbiome data and other omics data to learn from each other in a regularized framework. Then apart from the multivariate analysis, for instance, like motivated from microbiome data itself, we expect a lot of heterogeneity in the microbiome data, for which I have developed robust regression model or negative binomial co-sparse factor regression model. Next slide, please. Some of the recent problems that I've been working on or computational tools development is heavily focused on engraftment of the microbiome when it gets transferred from donor to recipient, or like causal model for the microbiome data sets. Those are some of the things that I'm heavily invested in, and currently a lot of tools I'm working on is on microbiome transfer from host to patient, which we are calling now mPRAC-X. So that will be up soon. And next slide, please.
I think the time is over, so just leave that slide, and we'll give you a. I didn't give a minute to the previous speaker, so we'll start with you, and there's a minute for potential questions. So we'll leave that slide up, and if there are any questions, please go ahead and do that. And if there are no questions, if there are any questions for our previous speaker, for Ravin, that would be great. Please feel free to ask questions.
I have my hand up. If
Okay, I'm
but
not
that's
seeing you. I'm sorry.
I'm
Okay,
sorry. Okay, hi, I'm Candace Floyd.
Hey,
So my
Candace, go
question,
ahead. Go ahead.
yeah, so my question's about the level of data that you need from a computational model perspective. And so what I'm interested in is trying to validate microbiome findings across species, from human to other species. So give me just kind of a sense of what types of data you would need to use that type of approach.
So you want to translate an understanding from one species to another?
Exactly, yes.
So some of the aspects so for instance, like some of the tools or the data set that we might need, it depends on the hypothesis. If your hypothesis is around microbiome as a species or microbiome as or if you're trying to understand microbiome as at the functional level and so on like that. So obviously, so from the genomics data, the count of the microbial species, if your hypothesis is around the microbiome data or microbiome data as in the species, and if at the functional level, then obviously some of the human output, like which focus more on the functional aspects of the learned from the genomics data, or like if you have 16S data, then we can use PyCrust as a tool, like to learn some of the functional aspects and so on like that. Some of those things could be so those are the two different types of data sets that I generally work on and try to develop methodology around it. So at which, like in some of my past work, like we have translated some of our understanding from mice model to the human model. That's what the approach that we have taken.
Thank you. Any other questions? Well, if there's no other questions, Dr. Floyd, you're up next. Candace is a recent faculty here at Emory. She's a professor in emergency medicine, and she was recruited in October of last year, of 2022, so about a year ago. And she is in the Department of Emergency Medicine, where she directs a translational neurotrauma research laboratory. So Candace, go ahead.
Great, thank you. So the main purpose of my wanting to be here today was to look for collaborators that might be interested in understanding the microbiome in some of these really clinically relevant models. So next slide. So we have been using large animal models of neurotrauma. And by neurotrauma, I'm referring here to traumatic brain injury, traumatic spinal cord injury, and models that induce neuropathic pain. So that could be a low back pain model is one of the models we use. It could be a nerve crush injury model. We use these porcine models of neurotrauma because they are complex physiologically. And they have anatomical similarities to humans that are much greater than those in rodent models. And that includes not only their CNS structure, their gray and white matter distributions, but importantly for this talk, it also includes things like the immune system. So immune system in pigs is much more like that of humans and rodent. And then the gut biology. So a lot of people think that pigs are like sheep. They're not, even though they are ungulates. They have monogastric stomachs, and they eat everything that we eat as humans. They share our same dietary preferences as humans. They have a gut physiology that is very similar to humans. Indeed, things like pharmacokinetics and pharmacodynamics in pigs are nearly identical to that of humans. Pig skin is nearly identical to that of humans, and so they're a really great model for human pathophysiology. Again, in terms of the spinal cord and kind of CNS anatomy, they also have a lot of similarities to humans. So next slide. So my team, what we have brought to Emory are these large animal models of clinically relevant neurotrauma and neuropathic pain. So I kind of joke when I say this because it's somewhat amusing, but it's a really valuable tool. My team speaks pig, so we use bioacoustic analyses to understand and interpret the pig's perspective of pain and distress, which is really important. If you think about a supraspinal outcome in a model, to understand pain. So we're not the only ones that think that pigs are a great model for success in translation and disease. Pigs have been successfully used in lots of other areas. You see artery disease, infectious disease, toxicology, vaccine development, skin grafts. There are genetically modified pigs. And as I said, they just have this really great similarity to humans. So we think of pigs as a translational intermediate between perhaps discovery research that can be done in some of the smaller animal or rodent species as a translational bridge to the human species. Next slide, please. So what are some of the tools that we bring and why study the microbiome? So again, I'm not a microbiome expert. I'm a neurotrauma expert, but there is emerging, that's what it is supposed to say, evidence that the microbiome really affects outcomes after brain injury, spinal cord injury, and really affects neuropathic pain. And the missing link, or one of the common pathophysiological links, may be microbiome effects on the immune response. So, and it seems to be this kind of bidirectional effects, right? So effects not only on the gut barrier permeability affecting brain, but also on the brain and the CNS injuries affecting the gut microbiota. So next slide, please. So what do we bring? Where do we want to collaborate? So we have really great clinically relevant pig models of traumatic brain injury, spinal cord injury, pain. We can do serial sampling of CSF, behavior pharmacology models, telemetry, image-guided neurosurgery, metabolic disease in pigs. We can do serial fecal collections, CSF collection, tissue collections. So we're really looking for collaborators that can help along this thing. I'll stop there. Maybe I won't get gonged. Yay!
Thank you. You didn't get conned. Great. Any questions for Dr. Floyd?
I have a question.
Yeah, go ahead and identify yourself.
Hi, Dr. Floyd. I'm Irene from Emory School of Nursing. I was just curious if pigs naturally experience neurological dementias like Alzheimer's, and if so, if the pathophysiology is similar.
The short answer is yes. And so the long answer is that pigs live a long time. And so because of the extended lifespan in pigs, some accelerated Alzheimer's models have been developed that, kind of can be better used in the timelines. But many of the pathophysiologies are the same, not only the brain-based pathophysiology, so pigs develop a lot of the plaques and tangles. They do that after brain injury as well. They're gyrocephalic brains, but also the cognitive deficits. So one of the other things I didn't mention is we're developing some touchscreen tests where pigs play video games with their snout for reward. And those are really excellent back-translated tests of executive function. And pigs with these brain deficits also show similar functions that patients with these brain deficits will have.
Well, thank you.
Any other questions? Well, if not, we will move to Dr. Yang. Irene Yang is an assistant professor in the Neil Hodgson School of Nursing, and looking forward to your presentation.
Thank you so much. So this is really fun, by the way. So thank you for putting this together for us. So my program of research really centers around oral health and thinking about ways to integrate it both clinically as well as in terms of research. And I have a vein of research that it's translational in nature, and I look at ways to integrate medical and dental care and ways to expand access to dental care and address oral health disparities. But for the purpose of today, I'd like to talk about the streams of research that I have that are focused on investigating the oral microbiome and inflammation as mechanistic pathways for us to understand what we know in the literature is a plethora of different kinds of epidemiologic studies that connect oral disease to extraoral conditions. Next slide, please. So why the oral microbiome? Dental caries, so cavities and gum disease, are two of the most common human diseases worldwide. And obviously, both of those conditions are closely related to the oral ecosystem, bacterial, viral, and fungal. The gut microbiome has gotten a lot of attention. It's the sexy microbiome, but I would argue that the oral microbiome is worthy of our interest and attention. It is a highly complex, in fact, the second most complex bacterial ecosystem in our body. The mouth represents the gateway to the human body. I mentioned before numerous studies that have connected the oral cavity, in particular gum disease, to various extraoral outcomes. We just don't know why that relationship is there, and the oral microbiome affords a way for us to investigate this. Also, the oral cavity offers matrices that are easily accessible for researchers like us, and then also potentially offer targets for diagnostic and intervention, for diagnosis and intervention. Next slide, please. And one more click. So I investigate this largely in 3 populations: maternal populations, looking at the relationship between the oral microbiome, oral disease, and pregnancy outcomes. I had a KO1 to characterize the subgingival microbiome in Black pregnant women and explore relationships with inflammation and preterm birth. And then I just recently was funded with an R01 to examine the contribution of the oral microbiome to Alzheimer's disease risk. And then I'm a co-investigator on an R56 looking at early gum disease and vaping. And I'm in charge of the oral microbiome, and we're looking at relationships with puffing topography. Next slide. I would love to talk to anyone who is interested in the oral microbiome here on Emory campus, interested in oral health in general. I think I saw something in the chat pop up, Kathy, and would love to talk to you. And what I need help with, I would love to connect with on-the-ground collaborators, clinicians, oral health clinicians, and/or researchers. And I've often bemoaned the fact that we no longer have a dental school. Would love it if the dental school reopened. Thanks so much. I think I beat the gong too.
Wonderful. You guys are getting good at beating the gong. I just want to notice that the 2 women beat the gong and the man did not. Anyway, any questions for Dr. Yang? Candice?
Yeah, so I'm wondering, forgive me for not knowing this, but has the oral microbiome been implicated in any type of pain-related symptoms? So I'm thinking of like trigeminal neuralgia, or that have this kind of dental to brain component. Can you think of examples where the oral microbiome has really influenced that?
That's a good question. I don't know about pain itself as a symptom, but there's definitely there's a hypothesized oral brain connection. And one of the hypotheses is that it's somehow nerve related. So that, either organisms themselves or something travel along the nerve pathways.
Yeah.
So I wouldn't be surprised if there is literature about a pain connection.
Hey, Dr. Yang, may I ask you quickly what type of methods you're using generally to study inflammation in your oral, washes or samples?
Yeah, generally I collect saliva samples. I have collected gingival crevicular fluid from my K, and then we obviously look at blood for systemic markers of inflammation. And then for the Alzheimer's project, we're looking at neurocognitive, a CSF for neurocognitive.
Any particular cellular assay you're using on the cells from the mouth itself, or you're mostly doing molecular markers?
No, we're looking at molecular markers, inflammatory markers.
Okay.
But I'd be open to.
Yeah, there might be some work that we can help with in that regard for sure.
Fabulous.
That sounds great. Well, thank you so much. Let's move on to the next presentation. And for the next presentation, I'm delighted to see my friend and colleague Solveig Cunningham, who is an associate professor in the Huber Department of Global Health, and somebody that I had no idea until today was actually interested in the microbiome. So Solveig, I'm great to see you. Maybe you.
Thanks so much for the introduction, Carlos, and thanks everybody for letting me join. Now, as Carlos is pointing out, I'm a demographer. Actually, next slide may give us a little bit more on that. Great. So so my background is in demography. A lot of my interests relate to obesity and chronic disease, as well as population level indicators of health over the life course. Over the next slide, please. So I have two really exciting project ideas that I've been mulling over and developing with, along over the past couple of years. And I was excited to see whether there are others in the group who may be interested in these topics. Topic number one relates to something that we see with foreign-born individuals moving to the US and other Western places. One of the interesting patterns is that migrants tend to be in better health when they first arrive in the US, but tend to develop obesity and diabetes, especially much more so than would be explained based on their age and even compared with the US-born population. So one of the interesting questions here, I think, given the changes that we know occur with changes in environment and changes in diet, there are likely implicated with migration, is are changes in the microbiome underlying some of these changing health risks among migrants. So as a very first step in this, I've worked primarily with students and done some both some primary data collection, really just on the survey side of this. The closest we got to microbiome on this was to ask people, would you be willing and interested in providing samples? And there was actually quite a bit of interest. We've also done a systematic review that was just recently published, finding that there is evidence of changes in the human microbiome with migration that may be implicated in changes in health and especially obesity along the way. Let's move on to the next slide to give you a little teaser on another related project. So one of the things you might not know is that most of us eat quite a bit of plastic during our day. In fact, the average American eats about a credit card's worth of plastic as part of our daily lives. Now, I expect that this probably has implications for our gut health that may be linked with metabolic health. Again, working primarily with students, I'm in the midst of a systematic review, trying to see what evidence do we have of this and what are the next steps here. So what we do see is that there's actually very little in this. In fact, there are no in vivo studies in humans. There's a little bit of research in other mammals that indicates that intake of microplastics is associated with changes in microbiome that could be along the causal pathway to obesity and diabetes, to be determined. Again, in humans, we have just a few in vitro studies, again, that are suggestive, but that leave a lot of really exciting opportunities to understand this further. Can we think of a pathway through which plastic may be associated with changes in microbiome composition, both in terms of diversity as well as in terms of specific species? Here I have the phylum level indication that would suggest that intake of plastics that probably occurs during our daily lives is associated with changes in species diversity. Over the next slide, please. Great to say thanks very much. Here are some of the ways in which I hope to find partners and collaborators. Thank you.
Well, thanks a lot. And I'm just tired of eating plastic, but you just shocked me. So, Ambar?
Hey, Solveig, excellent presentation. And fascinating research questions. So, when migration happens, there's a lot of change in nutrition factors. So how do you control for that when you look at microbiome?
Yeah, and it's all tied together, right? Because a lot of so we expect that there are going to be changes in diets, and we're getting pretty good at measuring changes in diets in many ways using survey data. Now, what the implications are for the microbiome, I think that's also testable, right? So we can look at what people are at changes in people's diets and potentially link those to the microbiome. And ideally, actually, we would do this right from before people even migrate. So the study that I'm developing right now would involve international students. So we know before they even come, right? We can even get data before they travel, when they first arrive, and then as they get established. So during that period of transition.
Awesome, fascinating. Thank you.
Any other questions? Well, thank you, Solveig. Oh, Bob.
Yeah, so how do plastics do this? I mean, what's the mechanism whereby plastics can have these effects?
So we're in the early stages. So we don't know a whole lot, but there are possible mechanistic pathways, and there are possible endocrine pathways. So the plastics themselves may have endocrine disruptor consequences, as well as creating blockages that may affect the gut. So that's the very big picture answer.
Can I make a comment?
Yes, please.
Go ahead, Roberto.
Absolutely.
Yeah, so actually 2 comments. First, many plastics are fairly potent estrogen, and as such, they can modify the composition of the microbiome. And this is an hypothesis that could be tested quite easily in mice. It would be simpler to do. And then the second comment is, should you be interested, I have microbiome sequencing data from two small study population that we have enrolled for a completely different study, one from the US and one from Switzerland. They are comparable for every other variable, and they have a striking difference in microbiome composition. So they don't plan to migrate, but if you need data about difference due to geography and diet, we have some data to offer.
Oh, wonderful. Thanks. I'll follow up with that, Roberto. Thank you.
Well, that's great. Well, thanks a lot. Thank you, Solveig. Our next presenter is Andrei Fedorov, who is a professor at Georgia Institute of Technology.
I hope everyone can hear me. So good late afternoon. I'm going to shift gear a little bit of this discussion. There was a wonderful brilliance of biochemists, biologists, the clinicians here, and I'm an engineer. So I'll try to speak the language common to this community, but I think we are able to offer some of the technological advances that can be useful across many problems that have been considered by this group. I was actually pleased to see that Rabin was talking before me, because we already kind of started the connection through a similar workshop just a month or so ago. In any case, what I would like to introduce here a technology that we have been developing. It's called Dynamic Sampling Platform. And its goal really to be able to measure intracellular metabolome, extracellular secretome, and surface markers, essentially membrane-embedded proteins, on the scale of individual cell. And what's important, at the same time and the same place. Essentially, what I would refer to is dynamic proteomics, metabolomics, and surfaceomics at the same time. You can see my contact information at the bottom, just in case you want to contact me. Next slide, please. So what I will do, will just briefly, you don't need to pay to this very busy, much attention to great details of this busy slide, but I would like to introduce each of this technology in turn, and I'll start with Dynamic Sampling Platform for extracellular analysis. Many of us would like to be able to measure at extremely low concentration, label-free, from very small sample volume, a variety of biomarkers over the broad range from metabolites to very large proteins that being secreted by either cells or microorganisms dynamically. And we are talking about sample sizes on the scale of few tens of nanoliters, few seconds to a minute analysis time. And the tool that we use is electrospray ionization mass spectrometry coupled to microfluidic devices that allows you to do this ultra fast, small volume sample prep. And the advantage that brings about is the ability to sample microenvironment from the cell. So essentially assess heterogeneity over the large population, track temporal evolution, be able to identify different cell types or through their secretome signatures, try to discover new secreted biomarkers and such. You can see a reference below for some example of this work. Next slide, please. Shifting gear to metabolome, we have just recently demonstrated technology that allows to measure intracellular metabolomes on the scale of just a few cells. We are talking about 10 to 100 cells that gives you a complete measurement of intracellular metabolite, amino acids, and other relevant metabolites from individual cells in such a way that it can be processed on the scale of few minutes, 10 minutes we demonstrated. The device is reusable and can be used for continuous monitoring. Next slide, please. This slide is an attempt to summarize essentially this, next slide. Kind of what I just said in the context of available state-of-the-art tools that you probably use, assays like ELISA, HPLC-MS, and this DSP allows you to measure secretome and metabolome from minimally invasive, real-time, broad spectrum, so it allows you to do endpoint and discovery work. And I'm gonna stop at 0.
Thank you very much, Andre. Any questions?
Well, I have one for Andre.
Yeah.
Andre, we discussed about host cells a lot. Do you have experience applying the same technologies to, fungi and bacteria? And I'm guessing, cell is a cell, but what's your experience with that?
Yeah, so far, all of our experience limited to working with immune cells, T cell, it's mostly T cells work that we've been doing here, but so no direct experience working with bacteria or other cells, fungi. I suspect it's not going to. I don't see, based on my limited understanding, fundamental challenges to why it may not work. The technology may need to be adapted, so some of the features of the technology may need to be modified. It's pretty much the characteristics of the cell, their size, membrane integrity, because we need to lyse the cells. We need to capture cells that depend on the mechanical property of the membrane, but it all can be embedded in the device design to be able to tune it for a given system, so to speak.
Thank you.
Yeah.
Andre, I noticed you've got minimally invasive mentioned there. Are you able to do this with samples taken from patients?
So the way in principle, let me just clarify. We do not have a system that actually, can extract sample from the patient. So someone will need to extract sample from the patient. Let's say it can be collections of cells, and then we can probe this sample locally. But we do not have a sampling interface that will be able to go into a patient and extract the samples.
Okay. Thanks.
I see there was a question there. No, I still have time there about extracellular vesicles. In principles, yes. The answer is, again, this will require an additional design. We actually started looking at extracellular vesicles as we speak. We there are some folks from UGA reached out to us who were looking at the exosomes and general extracellular vesicles and trying to probe its metabolome secretome. And so we actually have trying to start a project along this line? So the answer is, I hope so, yes. I think the question was from Kathy.
Yeah.
Do we have time for another quick question or not?
Yeah, go ahead.
Hi, fascinating. Have you done on the metabolome, secretome side of things, are you primarily doing targeted analyses, looking for particular biomarkers? Have you done untargeted? And do you foresee any issues with annotation and such if you were to switch to a bacterial or fungal cell contributing?
Excellent question. So we've done both targeted and untargeted analysis. We looked at everything from whole spectrum of metabolites, and the way you usually do it, it's starting with PCA to find principal components, then look at the loading plot, and then zoom in on a particular spectral feature to identify potential different metabolites that are involved, and then you couple usually with gene network analysis to see if particular metabolic cycle is involved. So it's kind of, there's a bioinformatic tool added to it, because just looking at the spectrum, you cannot easily identify a particular species. It's much easier, obviously, to do it when you know what you're looking for targeted process. You cannot compete with HPLC on the coverage. What you compete with HPLC-MS on the real-time analysis and the size of the spectrum. So in other words, if your problem is actually you suspect that what's important to be able to look at the time signature of event, your standard HPLC, which takes really multiple hours, days to run, and it's a very time average analysis, will actually fail to provide you, even on the targeted or untargeted level, an insight of what dynamically happening. And so that's the void we are trying to fill, at the expense, obviously, of comprehensive coverage of all possible biomarkers.
Thanks.
Thank you. Okay, let's move on to the next presentation. Next presenter is Dr. Taylor. Dr. Bob Taylor is professor at Emory and at the Department of Biomedical Engineering, and he's the chair of the Department of Cardiology, and he's also the PI for the CTSA.
All right. Carlos, thanks for elevating the division to a department level. We appreciate that. And we look forward to you doing that before you finish up your dean's. Yeah, so I'm a cardiologist and interested in vascular biology, and we've studied mostly hemodynamics and inflammation, and then we got interested in intestinal barrier function. So instead of thinking about the microbiome, we started thinking about how the microbiome could impact the rest of the body and how much of that was due to intestinal barrier dysfunction. And we sort of stumbled into this when we were studying sickle cell animals. And there's a bit of a literature about intestinal barrier dysfunction in a variety of cardiovascular disease, including hypertension, athero, and MI, and heart failure, et cetera. And there's actually some animal data that shows that the intestinal barrier dysfunction, the increase in permeability, actually occurs before the development of hypertension in several animal models and before the development of dysbiosis. Next slide, please. So, oh, that wasn't fair. That was 1 minute.
Oh, that was pretty quick.
Yeah. And so this is, we were studying sickle cell animals, and we noticed that they were sort of leaky everywhere. And so when we looked at intestinal permeability by giving gavage serum phytidextran, we noticed that they had an increased intestinal permeability. And next, please. And when we did some histology on that and some PCR and some Westerns, what we found is there was a lot of inflammation in the gut. And that bottom picture of the histology there is staining for neutrophils. And the top is just sort of a sampling of a variety of markers, TNF, IL-17A, and then CD68 is a marker of white cells, but just showing that there's inflammation in the gut. And this is mostly in the duodenum and the ileum. And next slide, please. Next slide. And, so because of this, we started looking at oxidative stress and the reactive oxygen species, and found that in the intestinal mucosa, there was an increase in superoxide in the sickle cell animals. And associated with that was a decrease in a variety of the antioxidant systems. Next slide, please. And just to show that this isn't all about sickle cell, if we take just regular old mice and feed them a high-fat diet, they actually get an increase in permeability too. And It's a more generalized mechanism, I think. I was actually just Googling microplastics, and apparently in marine animals, microplastics increase oxidative stress in the intestinal mucosa, which is interesting. Next slide, please. And so we've been thinking about it therapeutically, and our strategy has been to use a Trojan horse strategy based on the folate transporters that are present in the gut. And as you all know, folate is a required micronutrient. We don't synthesize it, and so you have two different carrier systems, mostly the proton coupled in the duodenum and the reduced folate carrier further down. And so we thought, could we put an antioxidant and conjugate it to folate? And that's what we did with Temple, which is a sort of a broad-based antioxidant, and conjugated it to folate. And on the next slide, click once more, you can see that, whoops, go back, please. There we go. So AA, so this is looking at intestinal permeability, and AA is wild type. And then you can see that when we give them the tempo folate, not much happens. Sickle cell SS has increased permeability, and that higher, even lower and higher doses of tempo folate normalizes that permeability deficit. And then if we give the controls of tempo and folate alone, they don't do anything. Next slide. And so what we can offer is we have mouse models of all sorts of cardiovascular disease. We have mice that overexpress catalase or antioxidants. We do a lot of vascular inflammation. And what I need help with is better methods to assess intestinal permeability in mice. Phytidextran is not great. And if anybody has animal models, as we develop some new antioxidants, because we're working on some new ones, we'd like to try those out. So that's what we're working on. Thanks.
Thank you, Bob. That was great. Any questions for Dr. Taylor? Dr. Floyd?
Yeah, does that differential sugar test work in mice? We do it in pigs, where we basically, then go back and look at the, different moieties of the sugar in the urine and in the gut. Does that work?
So people have not really tried it. We've actually just started looking at that and looking at some small fluorescent tag molecules, because phytidextran, as you know, is bigger than your sugars, and it only picks up a really big deficit or a really big defect. And if you have that assay up and running, we'd love to talk to you about it, because we've been thinking about that, and people have not routinely used it in rodents, and we're sort of wondering why, if it's just because they haven't thought about it, or if it's sort of just hard to do.
Yeah, I don't know. I've only done it in pig, but we could talk and see if it could be miniaturized.
We'd love to pick your brain on that. That would be fantastic.
Thank you. Any more questions? Okay, our next presenter is Ben Kopp. Ben is an associate professor in the Department of Medicine and the Division of Pulmonary, and he's the director of the Pulmonary Sickle Cell Program and director of the Pulmonary and Sleep Physician Research at Children's Healthcare Atlanta. So Ben, go ahead.
Thank you. All right. I'm a pulmonologist here, and I'm a lab neighbor to Ruben. So happy to on things moving forward. Next slide. So the focus of my lab is on sorry, I just got kicked from Zoom. Can you guys hear me?
We're gonna hear you now.
Okay.
Ben, why don't you turn your video off so your internet is working better?
Okay. Yes, sorry. So I am a pediatric pulmonologist, so I focus on kids with chronic lung diseases like cystic fibrosis and sickle cell. And our lab is really interested in how innate immune cells respond to the variety of pathogens, as well as environmental stimuli, and how they can sense and be rewired by these stimuli and influence their abnormal behaviors, both pro-inflammatory as well as dysregulated repair. And so we're interested in particular pathogens, that are classic with individuals like CF, but also the microbiome as a whole, and how that can influence disease over time. And particularly, again, with the changing environmental exposures that our individuals are faced with over their lifetime from early birth. Next slide. So one of the studies that I wanted to focus on is a new study in sickle cell that's involving the microbiome that we've started. So this is a longitudinal cohort collection of children with sickle cell that we are collecting at baseline health and at baseline yearly clinic visits, and then if they're hospitalized, we're doing a variety of biologic sampling to look at their airway microbiome and their gut microbiome and see how these change and are shaped over time. Our children with sickle cell are exposed to a variety of antibiotic exposures every time they have a fever, when they're hospitalized with issues like acute chest syndrome, and they're at risk for encapsulated pathogens. But it's not known if all these exposures are not necessarily appropriate at times of infection, because many of their illnesses are actually driven by viruses. So we're trying to capture what their baseline microbiome looks like. We have pilot data that's shown that they have an abnormal upper airway microbiome. And importantly, we really don't know what the healthy microbiome looks like in kids that are non-white. So we have a control cohort of race-matched children that we're collecting as well. And we're combining this with multi-omics analyses to look at airway signatures, blood signatures, and how their metabolites that are produced, their proteins and their inflammatory signatures are matching the microbiomes that are present. So I'm working with collaborators at the Hercules Exposome Center here. My collaborators back in Ohio State. So we have a cohort in Ohio and here in Atlanta to work on this study. Next animation. And so this is just early data, but we've been collecting oral and nasal microbiomes from healthy control individuals as HC, and then children with sickle cell disease. And we're getting pretty broad representation across different domains. Certainly highest in these samples is a lot of bacteria that we can then look at the specific abnormal taxa that are present. But we're particularly interested also in some changes in the fungiome that is present within these individuals, and some of the less well-known characteristics of the microbiome that we can detect in these samples. Again, looking at disease-changing states. In general, their nasal microbiomes seem to be more diverse than their oral microbiomes, but I think it's something we'll want to tease out a little bit further and potentially with some of the information presented earlier. So next slide. So I can offer to folks unique access to pediatric cohorts and samples, ex vivo modeling, like there have been some core resources within our pulmonary programs, but I'm interested in connecting with folks in this area more. Thanks.
Thank you, Ben. Any questions for Dr. Cobb?
I had a few questions, if I could. Really interesting, Ben. So I'm wondering what matrix you collect for the oral microbiome, if you do metagenomic sequencing, which I'm guessing you do if you are getting fungal identification, and then also how long of a period are you, like how frequently and how long are you collecting specimens?
Sure. Yeah. So we're doing right now, we're doing oropharyngeal swabs, so kind of posterior pharynx swabs, as well as nasopharyngeal swabs. So we were more interested in the connection with the airway from that side. It wouldn't be hard to add in probably a wash or something else as a potential. And yes, we are doing metagenomics sequencing for this. So they are all captured at baseline, and then we're doing yearly sampling. So the current grant, or the current IRB, is set out for about a 5-year sampling period. The funding for the first couple of years, and we'll be working to expand that to continue the longitudinal sampling, but at least plan for 5 years of sampling for individuals.
And do children with cystic fibrosis and sickle cell disease have oral kind of sequelae, like not respiratory, but specifically dental or oral?
Yeah, sickle cell definitely has some dental disease. My old clinic that I was in had a dentist that actually came there. I haven't seen one here, but they have some known issues. And in CF, they eat a high-fat diet, so they can often have some abnormal issues from that standpoint too.
Interesting. Thank you.
Hey, Ben, can you give us a sense of the ages of your cohort? How old are the pediatric population?
Yeah, that's a good question. Right now, they're more in the late adolescent and teenage years that we're getting to enroll. Just from a feasibility standpoint, as I set it up here, we're starting there. There's several swabs that are involved. We hope as it grows and develops, we'll be able to get more, start sampling some infants. But just as I'm new here over the past year, so just getting the infrastructure has been easier to go in that older population.
And the samples which you've taken it's the gut microbiome, the lung. Can you just where are you sampling?
Yeah, so again, oropharynx, nasopharynx, and then stool if they're able to provide for the gut. So we have a home kit collection that they can send us back for stool.
Good, and you have healthy controls?
Yes. That's what we're starting to grow right now is the control population. Some of them are matched siblings when we can. To control for the environment, and then we're going to be getting people from the Hugh Spalding clinic.
So Ben, like in terms of the environmental factors and pathogens, like what kind of data do we have? And is it like, are you hypothesizing that the environmental factors and the pathogens impact microbiome and which in turn impact your clinical outcomes? Is that how you are looking into?
Yeah. I mean, we know certainly from CF that things like secondhand smoke exposure, air pollution can impact their biomes, particularly their gut microbiomes are impacted by tobacco smoke exposures. So we think they are altering, some of the preferential growth of pathogens, but also certainly they can simultaneously impact inflammatory pathways. We have, with the geocoding and pollution modeling, we can get a lot of other external environmental factors to look at besides just like particulate matter in this study.
No, that's really great. Yeah, there are some recent methodology development around taking into account directional approach, two-way modeling, or like instrumental variable approach and things like that, which can account for even unobserved confounders. Which can be very suitable for your hypothesis.
Okay, yeah, it'll be great to connect some more.
Thank you.
Thank you.
Fantastic, that was great. Let's go on to the next presentation by Dr. Pacifici. Roberto is professor in the Department of Medicine. He is the director of the Division of Endocrinology, Metabolism, and an expert on bone disease.
Thank you. May I have the next slide? So we are bone biologists, and we are interested in studying how the gut microbiome regulates bone cells, bone growth, bone development, and the response to hormones that regulate the skeleton, such as estrogen and parathyroid hormone. And we have become very interested in identifying mechanisms of how the gut microbiome talks to bone, how the information is exchanged, something that we call the gut-bone axis. May I have the next slide? Next. So we have been very interested in estrogen deficiency and hyperparathyroidism because these are the two main causes of osteoporosis. And what we have found is that both estrogen deficiency and parathyroid hormone need the presence of the gut microbiome to affect the skeleton. Both hormones have no effects in germ-free mice or in mice treated with antibiotics. And this is because the gut microbiome regulates the differentiation of Th17 cells in the intestinal wall. These are very inflammatory T cells, very important for bone. And then driven by chemokine gradients, these cells migrate to the blood vessels and from there to the bone marrow, where they release IL-17, which is a very powerful bone loss inducing cytokines, and that causes bone loss. Migration of immune cells is also important in the mechanism of bone metastasis. We have done a study in melanoma bone metastases, and we have shown that in the presence of a healthy microbiome, the tumor causes the expansion of NK cells and Th1 cells. These cells, again, driven by chemokines, get into the bloodstream. From there, they reach the tumor size in bone, where they have an anti-tumor effect, reducing the growth of the tumor. But if in mice with depleted microbiome, there are fewer of these immune cells, there is a lower population of NK and Th1 cells reaching the tumor area, and there is a more rapid tumor growth and mouse death. So careful with using antibiotics in cancer patients. Next slide. More recently, we have investigated the process of fracture healing, and now we repair fractures. And this is an interesting story because fractures cause an increase in gut permeability. The gut permeability leads to the expansion again of Th17 cells, which are chemotaxically attracted to the callus, to the fracture repair site. And this time the Th17 cells exert a positive effect because they shorten the inflammatory phase of callus formation, leading to a faster repair of the fractures. In mice treated with antibiotics, there is a lower number of Th17 cells in the intestine, a lower number of Th17 cells at the fracture site, and it takes longer for the fracture to heal. Another important mechanism that regulates how the gut microbiome talks to bone is via the production of metabolites. Specifically, we have shown that certain probiotics increase the production of butyrate. This short chain fatty acid is highly diffusible, reaches the bone marrow, where it activates the formation of regulatory T cells. These T cells regulate CD8 positive T cells, which make Wnt/B. This is a Wnt ligand that activates Wnt signaling in osteolysis, leading to increased bone formation. Next slide. So, next slide, please. No, okay. Can you go back? Next. No, this is not mine.
Sorry, for some reason I got stuck. I'm gonna try to get back to it. I don't know why.
Okay. Go forward 2 slides. There's one more. Okay, so what we can bring to the table is a skeletal phenotype. We have fancy micro CT scanners. We have techniques to study the migration of immune cells from the gut to distant organs, could be bone or the brain or anything else. And what we are currently doing is studying on gender-affirming hormone treatment. That is the hormone treatment used by transgender people. We model this in mice, and that has revealed incredible changes in microbiome composition with this cross-hormone therapy, and we need help in interpreting metagenomic data, so analysis, statistical analysis of metagenomic data, and even just come up with a graphic presentation of this complex data set is a challenge for us, so we would like to get some help. Thank you.
Thank you, Roberto. Any questions? Well, there are no questions. Let's go to the next presentation. Madeline Hauser, who is an assistant professor at the School of Nursing. And Madeline?
Good afternoon. Thank you for the introduction. So as was said, I am a research track assistant professor at Emory School of Nursing. And in my research track position, my focus is primarily on collaborative projects with other investigators. I provide methods expertise, often for microbiome or metabolomics or multi-omics studies, as well as content expertise to advance projects across the School of Nursing. I also have some bandwidth to collaborate outside the School of Nursing. And next slide, please. So this setup means that I collaborate on a lot of different kinds of projects. So here are some examples of the microbiome studies that I've worked on in the past just few years. It includes studies of the microbiome in neurological conditions, occupational health, pregnancy and cardiometabolic disease, and then a large-scale multi-investigator center focused on microbes' contributions to symptom presentation across a range of chronic health conditions. So you see, I'm not necessarily married to any particular area of focus, but the focus is more on the methods. And so often in the microbiome studies that I collaborate on, the focus is not so much on the microbes themselves, but rather on the microbiome as a therapeutic target or a biomarker, or as a source of insight into the physiological processes to target for intervention, all with the goal of improving symptoms and health outcomes for people. Next slide, please. So what do I bring to these collaborations and what do they look like in practice? Well, I got into bioinformatics work from the biology side. I started in biochemistry and microbiology and then did my PhD in immunology with a focus on gut-brain immune interactions in Parkinson's disease. I have over a decade of experience working at the bench on both the microbe and the host side of things. And then I began to focus more on large-scale data analysis, statistics, coding, and handling omics data sets. So now I get to apply all of these aspects of my background to the collaborative work that I do. Next slide, please. So what does that look like for a microbiome project? Well, for investigators within the School of Nursing, thanks to the support that I receive from the school, I can serve as a resource to consult on microbiome projects, to analyze sequencing data, to help with omics data sharing, to repositories, etc. And this is great for, say, pilot projects that don't have funding to engage outside analytical support. And it also makes it easier for investigators who have a strong clinical or public health focus, but don't necessarily have that expertise in microbiology to explore a microbiome component in their studies. The ideal situation, though, for projects within or outside the School of Nursing is when I'm brought in as a collaborator from the development stages of a project. I often serve as a co-I on grants with a microbiome aim, and I assist with study design and put together data analysis plans, data sharing plans. And then when a project is funded, I can handle the data analysis as well as contribute to the interpretation, thanks to my scientific background. Now, currently, I primarily work with 16S data or the downstream analysis of metagenomic data that was processed elsewhere. But we are getting a computing cluster in the School of Nursing, so I'm hoping to expand my capabilities to processing metagenomic data when that happens. I'm also getting more into multiomics as well. So I wanted to talk to you today first because I think that Emory School of Nursing is doing something rather innovative in having a collaborative position like mine. And I wonder if that's the kind of role that we might see more of in the future. And then second, while I do stay quite busy, I'm certainly open to discussions of future collaborations, so feel free to reach out. And thanks for the opportunity to speak today.
Thank you, Madeline. Interesting presentation. Any questions?
Hey, Madeline, this is Rabin.
Rabin.
Congrats on your position. I didn't know that you are now faculty in the School of Nursing, so it's great.
Still around.
That's awesome. What's the opportunity that you have also to work on fungome data sets? I see that you said 16S. Are you looking also at analyzing some of the fungal data related to the microbiome?
So I haven't had an opportunity to do anything with a fungal data set in a number of years. Right now, the primary limitation in terms of metagenomics is just the computational resources that I have access to. So like I said, when I'm hoping when the computational cluster at the School of Nursing comes online, then I'll have access to that, and that'll make that a lot easier. But if there are, data sets that are processed externally, and then, I could work on the downstream side of things, then yeah, I'm open to that. And I have lab experience working with fungi and a lot of affection for the fungal side of things. So those are always interesting projects.
Hey, it's Candace. So my question is about the therapeutic screening or the therapeutic side that you mentioned. So do you have a screening platform for putative therapeutics, or do you have a favorite? You know, are you a fecal transfer fan, or, you know, kind of where are your thoughts on that therapeutic side?
So I don't have a drug screening platform or anything like that. Working with my collaborators in the School of Nursing, the focus tends to be more on behavioral interventions, diet interventions, things like that, or like I alluded to, using insights from the microbiome to point to particular physiological processes or metabolic processes or things like that that may be leading to symptom presentation or particular health outcomes that could be targeted either with drugs or with other kinds of non-drug interventions. So those are the kinds of projects that I more often work with. But in previous work more on the lab side of things, there was certainly an interest in drug candidates and things like that. But that's not as much the kind of projects that I get to work on these days, but open to it.
I have a quick question. I was wondering if you're also interested in analyzing mouse data set in addition to human data.
Yes, and there again, I have just a project or two where I've worked with mouse microbiome data, but that's mainly a product of where I'm working now. The collaborators I have tend to do more human studies, but like I said, in previous work, I did a lot with mouse models and have a lot of hands-on experience working with mice and such, and so I'm very familiar with the processes of microbiome sample collection and processing and that sort of thing for mouse studies.
Thank you.
Well, thank you, Dr. Hopper. Appreciate it. Why don't we move on to Reinhold Jones, who is the Emory Notobiotic Animal Core Director, and he's a faculty in the Department of Pediatrics.
Thank you very much. So I'm going to use this opportunity to share, to promulgate the services available to researchers across Emory at the Emory Notobiotic Mouse Core. So the mouse core, the Notobiotic Mouse Core, is located in the HSRB1. It's in the basement of the HSRB1 next to the specific pathogen-free animal facility. And this is what it looks like over here in the main picture. Inside each of those isolators are germ-free mice. So forward one. So inside an isolator like that, there are germ-free mice. The mice are fed germ-free food, germ-free water, and germ-free bedding. And we go through extensive testing to make sure that the mice are germ-free status. So the first service we offer to Emory investigators is to supply them with germ-free C57BL6 mice. So we have breeding isolators of CL57 Black 6 mice. So forward one. So also we offer a germ-free rederivation service in collaboration with Taconic. So perhaps you want to work with a knockout mouse and not just Black 6. So the way that we do rederivation of mice to germ-free status is that you send, and for example, in this example, if you wanted to rederive an IL-10 mouse to germ-free status, you send some breeding pairs from Atlanta to Taconic. At Taconic, they do an IVF into a germ-free Swiss Webster mouse. Then they put the germ-free mouse into a shipper, which is the yellow thing over there, right on the right-hand side. Then overnight transport of the shipper to Atlanta. Next slide. And next slide. And then the shipper arrives in Atlanta with pregnant females. We put these pregnant females into our isolators. Then the Swiss Webster female gives birth to your knockout germ-free mice, hoping that you have at least one male and female, and then you start a breeding colony from that. So germ-free re-derivation of mice we can do. Next slide. So also germ-free mice are germ-free, of course, but the real power of this system is notobiotics to know the microbiome within a mouse. So our workflow is to raise the germ-free mice inside the isolators, then we transfer them into these ISO cages, and then within the ISO cage we do a notobiotic study. Next slide. We handle these ISO cages very carefully and in a sterile environment and under a hood, just like you would cell culture. Next slide. And the utility of the notobiotic studies are to monocolonize a mouse with a single species of bacteria. We can also monoassociate a mouse with a bacterial-derived metabolite or agent such as short-chain fatty acid. We've done human microbiome associated mice, that is to take a human microbiome sample and colonize a germ-free mice with a human microbiome, thereby attempting to show causality in microbiome studies. And we've also done transient colonization, that is colonizing mice with auxotrophic E. coli that only survive for a short time within the mouse. Next slide. And then also we can collaborate with researchers to undertake specific techniques within germ-free mice. Next slide. So just to summarize, we can provide germ-free C57 BL6 mice. We can collaborate to rederive your knockout mouse to germ-free status. We can do specific notobiotic studies within the ISO cages, which are hermetically sealed cages, and we can develop specific procedures within the mice. We help with the ICOC amendments. We help with grant facilities documents. And that's the contact information for me, and this is our website. If you want to contact us, and there's 3 of us running the core at the moment, myself, Caroline Addis, and Amanda Metzger. And that's it. Thank you.
Thank you, Dr. Jones. Any questions?
Quick question for you, Reynold. Have you recolonized with fungi?
No.
Is that something that you could envision or not really?
Which fungi would you and
so
in what organ?
There was a paper by Ken Caldwell, NYU, showing that lab mice lack a fungo in the gut, and that is causing them to have abnormal myelopoiesis. That's why our lab mice tend to have a lot of lymphocytes and not a lot of granulocytes in their blood. He found that he could take lab mice, put them outside for a couple of months, or he could take lab mice and just colonize them with Candida. And then within 2 or 3 weeks, myelopoiesis would basically restart. And then, they would have 50% neutrophils, for example, in their blood, similarly to humans instead of 20%. So one of the ideas would be and now there's a new cell line. There's a new mouse line from Yale and another one from Japan, which they knocked in the GCSF receptor. So they're able to have chimerism with human myeloid cells. So I was hoping to basically combine the two.
So absolutely. So basically you could take a germ-free black 6 and colonize that with specific fungi, which you want. But then if you get hold of those specific humanized mice, you could rederive those to germ-free status. We could keep them inside the isolators, and then we could use the ISO cages to do specific notobiotic colonizations of those mice with your fungi or with any candidate agent, bacteria, or anything that you have.
Perfect. Thank you.
Fantastic. Well, thank you so much. We'll move to the next presentation. That'll be by Dr. Lyra Griffiths, who is the core director of the Emory Integrated Genomics Core and has been doing so for the last 4 years. So Dr. Griffiths.
Thank you very much. As this slide indicates, we have numerous NGS services at the Emory Integrated Genomics Core, but today I will be specifically emphasizing our services on 16S microbiome. Next slide, please. Our 16S microbiome services include a amplification of the V3, V4 region of the 16S ribosomal RNA. We provide DNA extractions for 16S microbiome profiling, and then also provide library prep. And this library prep is an Illumina-based library prep so that we can sequence on our MiSeq Illumina instrument. Data analysis services are offered by the Emory Integrated Computational Core, which is a partner core that can access data directly from our core facility. Next slide, please. And just to give you a little bit of information on pricing, we utilize the DNA Power Soil Pro Kit typically for our DNA extractions. This is a QIAGEN kit, and our DNA extraction and QC pricing is approximately $30 per sample. The best sample types that we work with are those that are a little bit more abundant. Those include oral swab, rectal swab, vaginal swabs, fecal pellets, and different types of soil. We can work with other agents or other samples that have lower abundance. It is important to randomize samples, just to go ahead and put that out there, between different experimental groups. We, as a core facility, offer specific controls. These include the mock community control. We specifically provide the Zymobiotics microbiome control. This is a combination of 8 bacterial and 2 yeast genomes. The 8 bacteria are equally distributed within that control, and the 2 yeast genomes at a different level, but they are also equally distributed. The DNA extraction negative controls, we also provide those so that we can just validate that nothing was introduced in particular from the extraction process. And we also provide library prep negative controls. These are all utilized by the computational core when they perform DNA data analysis. The V3, V4 library prep pricing is around $50 per sample for orders that have greater than 48 samples, and $68 for samples for orders with less than 48 samples. And then finally, sequencing of 16S libraries is, as I mentioned before, performed by an Illumina MiSeq. This comes out to approximately $40 per sample, just to give you a general sense of that. And so overall, if you were to perform all of these processes and workflows within the EIGC core, the price per sample is around $120 to $140. You can go to the next slide, please. And that was just a slide to say that we provide quality 16S library prep services. We would love to talk to you about any projects, and you can see that the EIGC website is on the bottom of the page, as well as our email address. And I'm happy to take questions if anyone has any.
So Lyra and Renata, I just want to make sure this is still true, that for our colleagues at UGA and Georgia Tech can use your services at cost of the Emory cost, is that right?
Yes, under the Georgia Research Alliance, any academic institution within Georgia can get the internal Emory pricing. And Renata, you can comment to make sure, is that true for your facility? Yes.
Yes, that's correct.
Thank you.
And any plan to do metagenomics?
We do not currently offer the metagenomics, so like shotgun sequencing at the Emory Integrated Genomics Core. We typically refer folks to Cosmos ID or other companies that do provide that, but we have heard some feedback that that is something that we may want to start to develop within our core facility, because those are relatively straightforward library prep procedures. So it's one of the things we're definitely considering adding to our repertoire.
Yeah. I think that there should also be a conversation about pricing, because Cosmos ID charges $135 per sample for metagenomics. So 16S should be cheaper. And your price come close to.
Sure.
Yeah.
I think that the thing that costs the most is the sequencing, and that's just something that with the price of the kits that we pay, it relatively kind of equates to the need to charge around $120 to $140 per sample. But yes, I understand that, like, if we were to also offer the metagenomics and it was about the same cost, that may be something that folks would prefer to do because they're getting more out of the library prep and sequencing costs.
Thank you.
Thank you.
Does the Corps also offer training opportunities for trainees, pre-doctoral and postdoctoral trainees, or is it really a drop off my poop place?
Sure. As far as the library prep and sequencing goes, we don't really have the capability to offer a lot of training just because of the amount of work that we have coming through the Corps facility. We do, though, offer a lot of planning projects, time to talk through on Zoom or in person, planning the projects to make sure that you have enough replicates for each experimental group, etc. Also just talking through like best practices for collecting samples. And again, Renault's also going to have those type of capabilities to talk through and expertise to talk through those. But that is something that, we do is kind of help plan projects. But as far as the training goes, it would be a specialized situation. So you can reach out and ask if there's something that we may be able to do. We typically do not provide that. Anything else? All right, thanks for this opportunity.
That's fantastic. Thank you so much. Very interesting.
So Lauren, maybe you could take the slides down during discussion so we can see each other. We lose Carlos? There he is.
Yeah, I'm here.
I don't know if you saw the note in Carlos, there was a sidebar going on in the chat about the post about maybe discussing more about the microbiome and pain. Candace, maybe you could start that off for us.
Yeah, I mean, we have a little bit of data where we have, sorry, my Zoom, am I back? Okay, we have a little bit of data that we did right before COVID. Unfortunately, we had early euthanized those.
Candace, you might need to turn off your video.
Yeah. Hold on, let me see. I think I stopped responding. Maybe you want to get someone else and I'll relog.
So Bob, you raised it. What specifically did you want to know? Well, Candace was getting back on.
Well, I never heard about that, and I thought it was a really interesting area, and especially with the HEAL funding initiatives out there, it seemed like it might be something more broadly for other people to think about, and sort of get out of their comfort zones and think about something a little different from what they've normally thought, and just appealed to me because of the sickle cell and some of the other disease states too.
Yeah. So Bob, am I still? Man, my Zoom is still bad. So yeah, I've reviewed recently for some HEAL R13 grants that are those really big, kind of like program project grants, and this type of collaboration would work really well. Looking at, kind of a centrally focused idea of microbiome across different pain states and various models. They do have to, there's another project that's a little bit more NINDS focused, but also would be probably that's that Combine RM1, which is, a large transformative ideas kind of project. So those might be some types of things that are in addition to your more classic P awards where you could come together.
Yeah, I think that the gut brain access and the transmission of metabolites and the connection with leaky barriers is really important in these processes. So Like I mentioned, yeah, I agree, Bob. Sickle cell would be a really interesting, model system for this. So I just connected Kansas with a couple other folks that are interested in that space from AFLAC.
So let me mention that Jessica Alvarez in our division, she's interested in cystic fibrosis and microbiome. She's a funded investigator. She also has expertise in metabolomics, so could be a great collaborator in this area.
There's a really good pig model of cystic fibrosis as well. It's been pretty well characterized. So that might be a nice translational tool.
Yeah, the CF pigs are super hard to work with, but certainly a possibility. So
I was going to say something sassy, but I won't say it. It's fine. We can do it.
Roberto, we're well connected with Jessica. She's part of our RDP, and we've been publishing several papers with her already.
Okay, great. Yeah. I personally was very intrigued by the microplastic story.
Yeah, that's really fascinating.
Microplastic, microbiome, and diabetes or obesity sounds very attractive area of investigation.
Can I ask?
If it's American Express or Visa, same effect?
How do you measure the amount of microplastics in a microbiome or in any other sort of fluid or tissue? Is Solveig here? Yes.
Yeah, I am here. Thanks for that. So all I can, again, I'm a demographer, so I'm coming at this from a very different perspective. Everything I have seen has been, as Bob was saying, in marine animals, and has largely been focused on the possibility of endocrine disruption through exposure. If there's interest, I can share. I have an early stage of a systematic review that's intended to give an overview of all this. So I can send that over. And I really think that this is an area that requires a lot more from expertise like those on this call.
I know very little about, but there are some key metabolites that can be measured in serum as a marker of plastic contamination. You can also really use histology to visualize particles, measure the size of particles in the gut. But by MS, it should be pretty straightforward.
I'm wondering, can you take a mouse and feed that mouse microplastics, sorry, and then detect them afterwards? Has that been done, or?
That's been a general approach, yeah, with a focus on
I think that the key variable is the size of the microparticles. So, and I think you can buy particles of any size you are interested.
Do we know anything about normal content of microplastics in, say, standard mouse chow or water or anything like that?
Well,
something I've never
I don't
thought about
know.
before.
It should be zero, but certainly it's not. I think that even newborns have plastic contamination in their tissues and bloodstream. So it happens.
And placenta.
It happens at conception.
Well, since mice eat everything plastic, it doesn't surprise me, right? So.
And now I want to add maybe the immunologist perspective to that. We have cells, macrophages, that are supposed to be taking care of debris during development and during normal life. And I think when we have microplastics in the system, we most likely overwhelm them through, they bind microplastics and they ingest them, and they might not be able to. They ingest them, but they don't digest them, right? So they get basically, full of microplastics. And part of, I think, the issue that we might be seeing with that level of contamination is that the normal role of macrophages in purposefully cleaning up normal debris in tissues is now being severely hampered by their being gorged by plastics. So we know already of some scavenger receptors that have affinity for plastics, like TIM4 is known. I think there's also some Siglex that have been shown to actually bind nanoplastics specifically. So that changes both the poise of the immune cells and their ability to do their normal job. So it's one of the reasons why I think you'll end up having, multiple comorbidities that are linked to plastic contamination and overingestion.
And so are there levels of contamination and overingestion that are known in the human literature? I guess I'm just wondering, it's kind of like a toxicity study, right? Could you back translate, levels that have been observed in certain populations to different animal models to evaluate pleiotropic toxic effects?
Not out there yet.
Well, I don't know the literature, but I would be surprised if it's not been done. I think in the environmental health literature, there's a lot of that. And there are a lot of study linking, plastic level to fertility, just to give you one example. And so I think that the novel angle is not so much what plastic do, but it's the involvement of the microbiome as an intermediary step between the plastics and the biological effect.
So is it that the normal interaction between microbiome and the cells that interface with the microbiome on our mucosae is messed up by plastic, and hence you have an effect on the microbiome, right? So you have the same basically mediator of interactions between microbiome and surface and plastic and surface. So that's the reason why you have maybe an indirect effect onto the microbiome.
Again, I have this endocrinology bias, but again, plastic, it's an incredibly potent estrogen. So if there is microparticles in the gut, you will have different permeability, you probably will have dysbiosis, you will have different metabolites. There are many, many mechanisms that could lead to changes in biological processes. Immune cells would be very much affected by estrogen-like substances.
Yeah, exactly. So the microplastic will directly affect the microbiome and also the immune cells at the same time. And then you finish off the triangle because the immune cells also want to sample the microbiome. So yeah, of course it's going to impact the that. I just did a Google search now, and there seems to be a few studies that have been done in 2022. So it might be something that's out there, might be worth a deeper dive.
Yeah.
So if there's enough interest in the microbiome and microplastics, we can put together another group for further discussion.
Yeah.
And it sounds like there are several of you who are interested. So if you would put your name in the chat, and Lauren will capture it, and we'll figure out how to move this discussion forward.
It's tough because we're using a lot of plastic in our experiments. We're putting our cells on plastic, we're growing our bacteria on plastic, we're making our media in plastics. So,
well, I can tell you an interesting story, but we sort of gave up studying the effect of estrogen in vitro because you can't have an estrogen-free control. The Petri dishes or whatever plastic you use, they have a very potent estrogenic effect. We had to put anti-estrogens in the control to sort of see the true estrogen-free conditions, and it's pretty scary.
a lot of interest popping up in the chat. So does anybody volunteer to lead that discussion the next time?
Could anyone give me some idea, like what kind of data we get when we are talking about microplastics or in microbiome?
So in terms of data, I think at the simplest non-invasive level, could be fecal plastic residue, but I'm sure there are other approaches also.
I'll be happy to take a look at the literature and report to the group.
Roberta, you just got elected lead for that group.
Well, I didn't say quite that. Let me see what I find first.
I know you didn't. I just volunteered you. We'll put the group together, but if you would start lead the discussion and solving, you started it, so we'll ask you to help.
I didn't start it.
No, Solveig started it.
Solveig did it.
So she can help.
Sounds great. Happy to.
Maybe there could be a online folder or Google Docs or something to share some of the papers too.
Yeah.
We can do that. Are there other sort of general themes that you heard during the session today that you'd like to expand upon?
I would say we had a couple of speakers talking about their skills in analyzing data sets and statistics, and Madeline and Adita, for example. Several of us needs help with data analysis. And again, I said during my talk, but I, for example, find it extremely challenging to generate the type of imaging that encapsulate, results of microbiome study. I had, for example, a statistician generated a 15-page long table for me, and I said, I can't put this in a paper on a grant. And the answer is, why not? You're a statistician, I'm not. A 15-page table doesn't work. So to translate that in a graph that can go into a paper on a grant is actually a big challenge if you don't know how to code in R or, come up with the tools that people in ecology or social science uses to describe complex interaction. It's quite a challenge.
Any comments from those who were observing but not presenting today? I can see from the names we have several people who are heavily into this field. So any comments from those watching?
Madeline, Adita, nothing to say?
She was asking for comments from those who didn't present, but no, you're absolutely right about the issue with presenting and summarizing the results simply. And, you just kind of hope that the journal you're submitting a manuscript to has a lenient supplement policy, and that's where you stick your 15-page table. But yes, trying to, I usually work in R, but if the issues with visualizing omics data, I mean, there are a few sort of go-tos with a few kinds of visualizations that are the go-tos, but it is an issue. How do you effectively summarize a massive amount of data while still making something that's interpretable and accurate?
So there is this recent paper coming from Curtis Hattonall's group, who's one of the pioneer in microbiome data, and this paper appeared in Nature. So maybe we can learn something about the visualization. So they basically did an integrated omics analysis with all sorts of visualizations. I think if certainly we do not want to publish a table or something like that, but we can certainly learn from already published paper, and they can give us quite good idea about like how they are presenting, how a journal like Nature is accepting or trying to understand the microbiome data for its reader and so on like that. But yeah, there are innumerable number of tools for even for visualization, but I think these papers speak itself. Like,
so I can tell you that the Harvard School of Public Health has an entire section of their microbiome center dedicated just to this. And we, you know, I think Emory would benefit from something like that.
More food for thought. Candace?
I'm skipping back to your question like 4 questions ago, sorry. And it was on another topic. And so I was wondering if my Zoom was misbehaving in the beginning. So wondering if we had collected list of folks that might be interested in microbiome and pain project. And then if we can take that to 2024 first quarter, I would be willing to kind of lead that next level discussion.
That's great, Candace. So if you're interested in the microbiome and pain, put yourself in the chat and we'll convene that group for you. And thank you for offering to lead it.
Thank you for that. After
2020, perfectly. All of this will be in 2024. I can guarantee it, because getting groups of people together is challenging.
Yeah.
But yeah, Michael Woodward, who's done a lot of this kind of work, has an interesting comment for you in the chat, Roberto. So we're just about out of time. I hope that those of you who are listening, those of you who presented, found individual collaborations. We'd like to follow up, because we like to, in order to keep offering these sorts of things, we have to justify ourselves, which means that we have to know whether any collaborations were made. So we will be following up with you. Lauren, you want to put those slides back up? We would just like your feedback on the session itself, whether it was helpful, if there are other ways that you'd like us to think about doing this. Gray put the evaluation link in the chat, so if you would fill that out before you leave, we would greatly appreciate it. So thank you for your attention today. A lot of interesting science.
This was really interesting. There's a lot of interesting stuff.
Yeah, I was just about to go back to you, Carlos, to see if you had any closing remarks.
I would just say that this is fascinating. This is a lot of potential opportunities of collaboration across not only the School of Medicine, but across the Health Sciences Center with people from nursing that I had no idea were doing such really interesting work. So I think there's a lot of really cool collaboration with the School of Public Health, School of Nursing, and advancing this field. And, you know, Solveig, you introduced me to plastics. It's a really bad thing. You know, Rwanda has banned plastics, so maybe we need to do the same here.
So Gray, can you fix the link? If not, we'll email it to everybody.
Yeah. Or if you, Kathy, maybe you guys can email the link to all of us.
Yes, we will. She put a new one in the chat, but we'll also email it, because we need the feedback.
Yep. Thank you very much.
Oh, thank you, everybody. Have a good rest of your day.
Thanks. Bye.
Take care. See you later.
Thank you. Bye-bye.